Welcome back. This is our second part of cellular movement, motility and contraction, and this time we're going to look at microtubule-based cell motility. So microtubules are crucial for movement of cilia and flagella, and those are the motile appendages of some eukaryotic cells. Flagella are significantly less common, and cilia are found on a number of cell types in a variety of tissues, and those cells have to move fluids across their surfaces. For example, the movement of fluid which moves a developing egg down through the oviduct or wafts mucus along the linings of the lungs. So cilia are about 2 to 10 micrometers long. They occur in large numbers on the surface of certain types of ciliated cells. They occur in both unicellular and multicellular eukaryotes, and they can be used either to drive the motility of an organism or to drive the motility or the movement of fluid over the surface of a structure. The cilia display an oar-like pattern of beating that's there, and that lets them generate a force that's parallel to the cell surface. So the cilia in a tuft will kind of twist back and then push forward. So if you think about it, that's the recovery stroke, and the power stroke is pushing that way. Then we have the recovery stroke, and then the power stroke pushing fluid. And so if the cilia is anchored down here, then we're looking at recovery and then power. So the force being generated that way is parallel to the cell surface. So that could move an organism through fluid, or it could move fluid across the surface of an organism, for example, across the surface of the gullet opening on a paramecium, for example. Flagella, they move cells through a fluid environment. You can imagine sperm swimming through seminal fluid, for example. They are approximately the same diameter as cilia. They're usually much longer though, up to 200 micrometers, so pretty long structures. And you only usually find one or a few per cell, and they move with a kind of what's called a propagated bending motion, and that generates a force that's parallel to the flagellum. And so here this sperm on the right would be swimming to the left, and so there we've got more of this undulating motion here rather than the oar-like motion that we had with the cilia. So it's two different kind of types of movement to generate force. Now both the cilia and the flagella share common structures, and that structure is generally called the axoneme, and the axoneme is connected to something called the basal body. So here is the axoneme here, that's microtubule based, and here is the basal body here, also microtubule based. And then there is an extension of the cell membrane which wraps around either the cilia or the flagella. So between the axoneme, which is this part up here, and the basal body, there is a transitional zone, and that's important because the microtubular arrangement down in the basal body is slightly different than the microtubular arrangement in the axoneme, and so this transitional zone basically transitions the microtubule arrangement from down here in the basal body to that which you see up in the axoneme. So the basal body kind of looks like a centriole. If you looked at centrioles when we talked about microtubule organizing centers, is what you have are 9 sets of tubular microtubules arranged as triplets. And so each of those microtubules in a triplet is sharing a common wall. And so those microtubules are called A, B, and C. And so the centrioles, or the centriole-like basal body, has A, B, C arranged triplet microtubules, and then we see them in part of the transitional zone, but then when we get up towards the top of the transitional zone, we see that the C microtubule has terminated, and we are left just with the AB microtubules and their shared wall, and then that will be the structure of the axoneme. And so this is a characteristic of 1 pair, 2, 3, 4, 5, 6, 7, 8, 9 outer pairs, and an inner doublet. So this is called the characteristic 9+2 arrangement that you see in the axonemes of flagella and cilia. So here we see, it's a little bit clearer here to see the 9+2 arrangement. So 1, 2, 3, 4, 5, 6, 7, 8, 9. We can see here using this transmission electron microscopy through a cross-section, a transverse cross-section through a flagella. And then here is the central microtubules. Now the pair in the center are singlets, whereas these on the outer, around the outside, are doublets. And you can see again here our A tubule and our B microtubule. The A microtubule here is the complete microtubule, so that one there is complete, and then the B microtubule is incomplete, so it doesn't have a complete set of 13 protofilaments. It could be 9, 10, or 11, just depending on how it gets synthesized. And then these two in the middle are complete microtubules with 13 protofilaments. The microtubules here also contain a protein called tectin, and the tectin's found at this kind of shared wall here. And that's kind of related to an intermediate filament protein. So just be aware there's some other proteins having some functions here in this axoneme. If we look at the diagram here, we can see a lot of the other proteins that are involved in the axoneme structure. Each A tubule, so that's the complete ring, has one of these radial spokes associated with it, and you can see then there is also a side arm, and then there are side arms of axonemal dynein. We talked about dynein before, and we talked about primarily cytoplasmic dynein. This is axonemal dynein, slightly different, structurally different. And it's the axonemal dynein which is involved in the sliding of the microtubules against one another, which leads to the bending that you need to get this motion or the undulation in the flagella. So the dynein motor protein is going to be responsible for the flexion that we see. The dynein arms are occurring in pairs. There's an outer arm and an inner arm. And then we've got these radial inner spokes here, and these set of spokes which are radiating in towards the central pair of microtubules, they are largely responsible for the force generation. The dynein generates the flexion, and then the force of the bending is produced by these radial spokes that we see in here. So let's take a look at the mechanism for the bending here. First thing to note here is that the overall length of microtubule doublets, whether you're talking about cilia or flagella, does not change during a beating cycle. Instead, is what happens is adjacent outer doublets slide relative to one another, and so they're sliding relative to one another rather than sliding like this up and down. They're sliding relative to one another, and that sliding movement is converted to a local bend. Then the radial and the circumferential connections restrain the sliding, and that's what leads to the bending. Now if you take out some of these linkages, like the Nexin linkage, and you allow the dynein to do its motor activity, is what you'll get the doublets sliding past one another rather than the flexing that you see, which generates the undulations that we talked about already. So the dynein provides the driving force, I guess, and if you remove those, then you're not going to get any flexing at all. So if you take the dynein out, you don't get flexing. If you put it back, you get flexing again. So it's the dynein which is absolutely responsible for generating the force required for axonemal flexing. A kind of an interesting question is how do you build a flagellum or build a cilia, because you've got to get tubulin to the tip, and presumably you've got to get other things back from the tip. So tubulin subunits are shuttled to and from the growing flagellum tip by these plus and minus end directed motor proteins, and this is called introflagella transport. So this is the movement of material up and down a flagella or a cilia. So kinesins, these two proteins here, they're going to move material towards the plus end of the flagellum tip, and then we'd have base directed movement powered by dyneins down here. And so we can move material up and down the axonemal filament so that we can grow and shrink the flagellum as we need to. Okay, that's it for microtubule based cell motility. We'll come back in a moment and start looking at microfilaments and the motor proteins they're involved with. I'll see you soon.